Thank you very much, Laurie, for introducing me, and thank you very much for having me today. I'm going to start out with a very simple problem, namely how to find the position of a fluorescent molecule, of a single fluorescent molecule in the focal plane of a microscope with maximum accuracy, precision, and speed. This is a very simple problem, but you will see it has many implications. Now, the way it was done actually over decades was the following. Let's assume we have a molecule sitting somewhere within the focal plane of a microscope, and we need to find out where it is. And then what was usually done is that you illuminate the whole field of view at the same time with light that produces fluorescence, that excites the molecule. And then, of course, the molecule will emit fluorescent light, and the fluorescent light is projected onto a camera. Now, the camera, consisting of many pixels, will actually produce a blob of diffracted fluorescent light here in the field of view of the camera. That will scale, of course, with the wavelengths. Now, how do we find out where it is? The narrative goes as follows. It's while this blob is very blurry, it's no problem to find out the exact position of the molecule, because that must coincide with the maximum of the blob here in the image plane. Now, if you look at the maximum of the blob, of course, that also has a certain uncertainty, because this uncertainty, of course, is given actually by the brightness of the fluorescent blob. And then, if you do the simple math, you will find out that this localization precision, of course, not only scales with the wavelengths, but inversely with the square root of the number of detected photons on the camera. So, with other words, the more photons you have on the camera, so the more photons the fluorescent molecule produces here, the better you will find out the position of the molecule within the focal plane, because this position on the camera directly correlates in space with the position of the molecule in the focal plane by simple geometrical optics. Now, this kind of concept, of course, has been used in many, many biophysical applications when it was important to find out the position of a fluorophore, and of course also in so-called single molecule-based super-resolution fluorescence microscopy. However, this concept has a major problem that comes clear actually from the fact that you have this dependence on the number of detected photons. You need to maximize the number of detected fluorescent photons. And if you need to maximize that, of course, especially in fluorescence, the issue is that fluorescent molecules, they tend to bleach, so meaning they go off after a while, so they don't emit any fluorescent photons anymore. What is even worse, they blink, meaning that they go off and then they stay off, you don't see them anymore. And of course, if you need to maximize the number of counts, the method is inherently slow because it takes a long time until you have them. If you're a physicist or a physical chemist by training, then you will also appreciate the fact that not only this takes a lot of time, there's also another fundamental problem, because molecules are, of course, dipole absorber or emitters. Say they are oriented like this, for example, and then they cannot emit in this direction or in that direction along the axis. They must emit in an axis that is kind of perpendicular to that axis of the dipole orientation. And that means that the pattern that is actually produced gets tilted to the side. So in other words, while you might get that position with high precision, it might be in the wrong place, because in fact it's 5 nanometers, 10 nanometers, and in the worst case, even 100 nanometers away from where you think that it is. Now, this has kind of made me think about another way of finding out the position of a molecule. And I came up with a different concept, which in retrospect, I must say, clearly was one of the best ideas I had in my life. And I'm explaining to you how it actually works. Now, instead of illuminating the whole field of view, what I decided to do is to use a donut beam. Now, a donut beam is actually a beam of light that is converted from a normal, say, Gaussian type of beam into something that has a center here that is 0 in its intensity. Now, why is this interesting? This is interesting because the 0 of intensity can perfectly define the position here, or a certain position in sample space. In other words, the position of the 0 of a donut beam is perfectly defined because this is exactly the place where there is never ever a laser photon showing up. So if you go slightly to the left or to the right, there will be some photons showing up according to this graph that we have here for the donut. But here at the center, never ever there will be a photon showing up. So this is perfectly defined. Not only that, it's possible to shift that donut around in sample space by a beam deflecting device, so a galvo or electro-optic device, whatever. So this position is perfectly known. You can argue with picometer precision. What is unknown, of course, is the position of the molecule. Now, that position can now be found out by finding out what is the distance actually between this and this. Because once you know the distance, once you know where the molecule is with respect to the known position of the donut zero, you have found out where the molecule is, and the localization process is, of course, sorted out, and the job is done. So how can this be done, and why does this work? It's very simple. If the two coincide in space, say the donut zero is just on top of the molecule, what will happen? There will be no excitation. Yeah? Note that this is now excitation light. So if you know a bit about stat, it's not light to turn fluorescence off. It's light for producing fluorescence. If the two coincide in space, then there is no fluorescence measured here at the detector because there is zero excitation intensity at the donut center, and so the molecule will not emit. Here, again, I mean, here there is substantial fluorescence produced by the intensity that is found here at the donut crest. Now, you can say that the brightness here that we measure here at the detector will tell us how far the molecule is away from the donut zero. If it's zero, it's just on top of it. If it's slightly away, there is some intensity. It's further away. There's of course, much more intensity and therefore much more fluorescent light. Now, why is this concept radically superior to what you've heard before? In order to make this clear, and with my last name being Hell, I decided to come up with a demon. And this little demon, of course, knows something and can do something that we humans cannot do. And let's assume for the moment that the molecule starts moving. Now, if the molecule starts moving like this demon, within an attosecond, and attoseconds have become very fashionable lately, within an attosecond can put the donut zero just on top of the molecule. So if the molecule moves, the demon instantly puts the zero on top of the molecule. And so in this case, of course, the demon can trace the position of the molecule with arbitrary precision, picometer precision, because it knows, the demon knows that, well, the molecule must coincide with the position of the donut zero, which is perfectly known because he is in control of it. Yet there is no fluorescence emission taking place. So in other words, this thought experiment tells us that at least in the limiting concept, so to speak, of a thought experiment, of a demon, we can perfectly trace the position of a molecule with arbitrary precision, picometer precision, without requiring any fluorescence emission, so zero fluorescence emission. So this is quite the opposite of what you've heard before. Now, there's also another striking consequence of this. There is no wavelength dependence. So previously you've seen it scales, the precision scales with the wavelengths. Here, the precision won't scale with the wavelengths. And you argue, now, why is that? Because, you know, I'm a physicist, I learned that if you have propagating fields of light or any waves, everything that you measure must scale with the wavelengths. But here it's gone. The reason is very simple. Well, in the end, the molecule is something like a point. And of course, the donut zero is something like a point, yeah? It's a singularity. And what we do here is we match 2 points. And therefore, it doesn't really matter what the wavelength is. So here, the donut zero could have made with green light, yellow light, blue light, red light, microwaves, X-rays, whatever. In the end, we just care for the point, and therefore the dependence of the wavelengths is gone. And then, of course, also important, the dipole orientation effect is gone. Why? Because if you minimize the fluorescence, that hardly matters. So without going into details of that, in reality, of course, there is no such thing as a demon. So you have to come up with something that works in practice, and that comes as close as possible to the demon case. And that means that we replace the demon with an electronics, a controller that counts the number of fluorescent photons that will now show up, because if the molecule moves, of course, no electronics will be able to push within an attosecond the zero on top of the molecule, but it will take some time. But the electronics can be programmed such that depending on the brightness that you get here on the detector, the zero can be placed on the position of the molecule. And the better the whole, say, closed loop works, the fewer photons you need in order to find out where the molecule is with a certain precision. So this is the idea behind this concept called MINFLUX. So we try to come as close to the demon situation, and the consequences are, of course, quite substantial. Now, previously, you've seen that sigma, so the precision with which we find out the position of the molecule with respect to the donut zero, scales with the wavelengths, but now this is gone. the wavelength dependence is gone. It is replaced by the average residual distance between the molecule and the donut zero. So the smaller you make this distance, yeah, the smaller the sigma will get, the higher the precision will be. And so this wavelength dependence is gone, and in essence, we are in control of making the precision much better by making this distance on average much smaller. Now, how much can we get in practice if you make L as small as you can practically do? So at the current stage of development, it's possible to get the same precision with 100 times fewer detected photons than with a camera. And that can be used, of course, now to follow something with a speed that is 100 times higher, or alternative square root of 100 is 10. You get the same precision with fewer photons, or of course, you get 10 times more precise if you have a certain number of photons. So you can use either advantages in order to find out something out. Now, especially this kind of quick localization is very appealing because this tracing of fluorophores has been used in biophysical experiments for 4 decades, I would say. And this is arguably one of the most famous cartoons in science, where the kinesin walks actually on a microtubule. And how was this walking found out? It was found out actually by making biophysical experiments where a bead is attached in an optical trap to the kinesin. And this way stepping was seen. Then another way of doing it is to use a fluorophore that is attached to the head or feet of this kinesin molecule. Both have their advantages and disadvantages. The disadvantage of using a big bead is, of course, it's very large. It's 300-400 nanometers in size, and of course, you cannot be sure whether this tiny kinesin is just attached to that bead, or maybe there's more kinesin attached to it. And of course, the fluorophore has the advantage that it's minimally invasive in as much as it's very small, 1 nanometer in size. But it took the people who did those experiments, Paul Servin and Ahmed Yildiz, actually 350 milliseconds to get a precision of 1 nanometer that is required to see those steps. And so, they saw this kind of humanized type of walking. And of course, you never know if that is true or not. We applied MINFLUX because we knew we would get that precision of 1 nanometer within much less time, say 5 milliseconds. And then, of course, you can attach it either to the stock or you can attach it to the head and then redo the experiments again. And this is actually quite interesting because you can see now, due to the higher spatiotemporal resolution, you can see details that you couldn't see before. And one of the advantages that the method instantly brings about is that you can see longer traces. So this is time, and these are the steps. And you see, due to the, say, high speed and temporal precision, you see also substeps. So substeps are steps that are actually not really anticipated from the cartoon. There are steps in between. They were seen actually with beads, but it wasn't clear whether they are true. We could see them for the first time now directly in a trace, because we could see, we could get this high spatiotemporal resolution. So this is this distribution of the steps here of the microtubule of the kinesin on the microtubule. Now, because we are faster, we could do these measurements at a higher speed of walking. So these initial experiments, which were done 20 years back, because they used a camera, they had to reduce the ATP concentration. So that's basically the currency of energy. And then, of course, if you don't reduce the ATP concentration, then you're too fast as a kinesin. When the kinesin walks, then you cannot localize the kinesin quickly enough. But because we have this higher, 100 times higher spatiotemporal resolution, we could apply, say, physiological ATP concentrations up to 1 millimolar and see the walking actually taking place at its own pace, so to speak. Currently, we have indications that this humanized walking is not the full story, and we are investigating this, but you can still argue in these experiments, this is done with microtubules, and the kinesin walks on microtubules on a cover slip. This is the way it was done for ages, but it's better to do it right in a cell. And I think this is actually the future of it, because this concept, MINFLUX, because it doesn't use a camera and has a pinhole in front of a detector, allows it to go straight away into the crowdedness of a cell. And so the next step in the development was to really prove that we can follow the movement of a motor protein, in this case dynein, as it works in a cell. And there is no argument about whether this is physiological or not, because this is in a living cell. You have all the other proteins around and the rest of it, the right ATP concentration. And this has allowed us to see for the first time, observe dyneins minimally invasively or basically non-invasively just by following the fluorophores on the dynein. Now, there's a discussion of whether there is a so-called tug of war between kinesin and dynein when they switch between moving away from the nucleus to the periphery of the cell or back. We have seen that very rarely. This is such a trace where such a tug of war was seen. And on the contrary, what we've seen very often is a rapid switch actually between this retrograde transport and anterograde transport that takes place within the duration of a single step. So what I'm saying here is basically the fact that we have a higher spatial depth resolution allows us to readdress this motor protein business again and see things that couldn't seen before because the spatial depth resolution is higher. Now, I'm shifting gears. I've been working, as you know, on resolution for quite some time, and there were papers 3-4 years back that said, if you're below 10 nanometers, then this whole super resolution thing will not work anymore because that's too close of a distance. It's true that if you want to measure, say, the distances between two fluorophores, what people usually thought about was to use a methodology called FRET. They said, okay, if I want to know if two proteins colocalize that have two fluorophore attached to them, I'm using FRET. Now, FRET is a method that doesn't give you the distance directly, it gives you that distance indirectly. So you have actually two different types of fluorophores, a so-called donor that has a shorter absorption and emission wavelengths, then the energy is directly transferred to a so-called acceptor that emits at a longer wavelengths. Now, this dipole-dipole interaction, which is heavily used in many, many biophysical applications, has a big caveat. The caveat is, again, the orientation, because the distance that you infer actually from the efficiency of this energy transfer depends on the orientation of the molecules. So if they are oriented unfavorably, then the distance may appear large, although it is not. So it's kind of inaccurate. In addition, of course, this distance is inferred from a law, actually, that scales with the 6th power, actually, of the distance. So the energy transfer scales inversely with the 6th power of the distance. So it's a very nonlinear function. So since I worked on resolution, of course, the question is, can you measure the distances directly? Say, if it's below 10 nanometers, can you just take a picture and say, this molecule is here, the other one's there, I subtract the two positions that I found out, and then I know what the distance is. And of course, MINFLUX should allow you to do just that. Now, at this point, I'm making you aware of the fact that in order to separate two fluorophores at sub-diffraction scales, the key physical enabling element so far was the fact that molecules were modulated in their ability to emit. Yeah, you play on-off. So in other words, when this molecule is emitting, the other one has to be silenced because otherwise the two are confused. And when this one is emitting, this one has to be silenced. And so this on-off transition is critically needed to discern two molecules at sub-diffraction distances. And this is the key physical enabling element behind STAT, PALM, STORM, PAINT, SOPHY, all the super resolution methods that have been developed until now. The role of the STAT beam is to shut off fluorescence. PALM, you need a photoactivation. Yeah, STORM is the same thing. PAINT, you need a binding or unbinding process. So let's do that now with such an on-off transition, and let's do a kind of experiment that is classical for the FRET type of research. It was done in the 60s. Namely, in order to establish FRET, Hoagland and Strickler, at that time, they decided to vary the distance between two fluorophores, the donor and acceptor at that time, and see how well FRET can measure actually the distance between two fluorophores. But now we don't use FRET, we don't use two fluorophores, we use fluorophores of the same kind that can be activated, so turned on and turned off. And now we have a kind of ruler, depending on the number of prolines we have here, and then we measure the distance actually between the two identical fluorophores that are sequentially turned on and off. Okay, now this is the measurement done with MINFLUX, and you see here that it works very well. So these are examples, 3.9, 5.1, 6.5, and so on. And here the smallest one is ladder 2.8 nanometer distance with 3 Angstrom precision measurement. This is the Fred range. In fact, we can go beyond the Fred range. That's very clear. Why not going higher up? It's just measurement of distances. And this clearly shows that because of the high precision that we get, with MINFLUX, we have the ability to directly measure the distances between molecules that are very, very close. And this is actually showing an example which I think is quite astounding. So I have a collaborator in the institute in Göttingen who works on NMR, and he's interested in this homodimer. And he knew that the binding pockets actually for fluorophores in this case are very close. So 11 Angstrom, 1.1 nanometer. And the question was, can we discern them? Can we measure the distance? Here it is, 10.9, so 0.6 Angstrom precision distance measurement. Okay, does it get closer than that? No, not really, because you know the fluorophore has about the size of about 1 nanometer, and basically it means the two fluorophores are touching each other, literally touching each other, yet we can discern them, because as I said, we switch on and off sequentially, so when this is emitting, the other one is not, and vice versa. But of course in projection, if this is kind of tilted, then the distances can become smaller, and you can measure them, So 6.6 Angstrom, 3.1 Angstrom, sub-Angstrom precision goes down basically to nothing. And this really shows that what people call super-resolution fluorescence microscopy or nanoscopy has really come down in resolution to basically molecules touching each other and basically down to nothing. Now, all done with focused light and objective lensing, and for me it is very satisfying to see how far this journey has actually taken us, basically down to molecular scale. Now, I could say now I've done my job, I do something else, but there's one thing that really bothered me, I must say. I said it many, many times. The key physical enabling element of STAT, palm, storm, paint, and the rest is on-off. You separate by making sure that not both are on at the same time, or 5 or 6 of them are on at the same time, but only one. And I repeated that hundreds of times in all the talks I've given since, I don't know, 2000 and something. In fact, I also stated it in the Nobel banquet speech. I had the honor of giving the banquet speech for the chemists, and I said to the king and the queen that the key element is the on-off transition and the excellencies and so on. And if you say that so often, you think, maybe there's still a way out of it. And if there's a way out of it, let's see if there is one. So the question is now, is this really needed? Is this really absolutely a must in order to overcome the diffraction barrier? So let's do the experiment again. Let's revisit the so-called Rayleigh criterion. Let's keep both molecules on. So there's no switchable compound, no activatable compound. You take the best fluorophore that you want, and it's just a fluorophore, okay? You put it over here. Now, why did people say this doesn't work, separating molecules that are that close? So here they are very close, much closer than the diffraction blob that is produced by, say, a microscope in the focal plane of a lens. Say this is a confocal microscope, you have this blob of light. It's very simple. If you scan that blob of light across the two molecules, you will get a kind of Gaussian spot, and you cannot tell these are two molecules because this still looks like a single spot. So if the two were at the same place, and you scanned again across it, this looks like this. There's hardly any difference. If you subtract the two, in fact, there is a subtle difference, but it's basically confounded by noise. It's all noisy, this difference. If you look into the profile, this is the profile here of the yellow one is the profile here on the left-hand side, the black one on where they do overlap, and you see there is a difference between the two that is completely within the noise band. So there's no way of discerning the two molecules because you cannot separate this situation from that situation. So this doesn't work. But let's redo this experiment now with MINFLUX setting, with a donut. Okay, so in this case, where we have a donut for excitation, I'm doing it again. If we scan, of course, with a donut over the two molecules sitting in the same place, we get a picture that looks like a donut that has a 0 here at the center. Why? Because if the zero here of the excitation light coincides with the two molecules, the center must be zero. There cannot be any fluorescence light detected. But what happens if the two are slightly apart? If they are slightly apart, no way that there is a zero here, because when the zero of excitation light coincides with this molecule, there will be fluorescence from the other molecule, and vice versa. When the zero coincides with that molecule, there will be fluorescence from this one. If the zero is in between them, both will emit light. So in other words, here at the center, no way that there is a zero. If you make the subtraction now, you will see, yeah, this is a measurable difference. So I can decide whether it's this case or that case, because there's a measurable difference. And if you look at the profile, of course, now the profile of the two where they're separated is clearly out of the noise band. Clearly out of the noise band. So the black one is the one where they sit in the same place. The orange one is the one where they are apart. In fact, it's even more crazy. Not only you can separate them, the closer they get, yeah, the better you can separate. And this is kind of mind-boggling, because psychologically you might think, okay, if you want to overcome the diffraction barrier from going from 200 to 100 or 50, the closer it gets, the harder it is. But if you do it the other way around with the minimum, the closer they are, the easier it is to separate. So 5 nanometer separation in principle is better than 50 nanometer separation if you do it with a donut. And the question is, does it work? And the answer is yes, it does. Here there is separation. Here actually done within a controlled sample where we knew exactly what distance we have applied, and this is the measurement. You see it goes down here from about 40 down to slightly below 9 nanometers. So this is a clear measurement where we separate two molecules that are constantly emitting. There's no on-off. They're constantly emitting, and still they are clearly separated. So why is this important? Not only because they separate 2, you can also separate 3, also as it is shown in here, or even 4, and of course also in 3D. So it's not just the fact that you separate 2. So it's a very general concept, as long as you are staying close to the minimum, so to speak, as long as you are at the true nanometer scale. Now the importance comes out instantly, because if you have on-off, yeah, then it means that you cannot see this molecule when the other one is emitting, and vice versa. But in this case, of course, you can trace all the molecules constantly without any interruption, and you don't have to use a blue and a green and a yellow one to separate them. You can separate them and still see them moving. And that means that you can take movies, so to speak, of constantly moving emitters all the time. And I think this has important implications, because what you can imagine now is to do a following experiment. So this is a protein, yeah? It's not a specific one in that sense. And of course, we know now protein structure, we know it from X-ray crystallography, NMR, cryo-EM, and AlphaFold by now. But it's not so easy to understand the function mechanistically. But what we could do now is we attach this fluorophores basically to 3, 4, 5 points, yeah, of the same color. You can add also other color if you want. There's nothing wrong with that, and watch them move. And then we understand how the protein works. So what I think, whereas super resolution microscopy told us actually where proteins are in the cell, MINFLUX has the potential to show us how they actually work mechanistically. And this is something that really excites me and also excites the people in my lab. Everyone wants to work on this and not on other things anymore. I think this is a lot of fun right now. There's another avenue coming up. So far, all this subdiffraction imaging was fundamentally limited to fluorescence. And you ask me why. So when I looked into the problem, I actually wanted to break the diffraction barrier in general, not just for fluorescence microscopy. There was no particular reason. I was a physicist. I had nothing to do with bio or so. And then I figured out that overcoming the diffraction barrier is easier for fluorescence because you can play with states. And that was arguably my first big idea. I realized that if you play with the states of the fluorophore, then you can do something that you make them separable. Now, here, we didn't have to do this anymore. It means now that we can apply this methodology to anything, yeah? Need not be fluorescence, as long as you have point objects and they stay very close. So I think transferring this to Raman scattering, other types of contrast is definitely viable. So this is something that is coming up. So another new avenue that's coming up. And with that, I'm thanking you very much for listening so patiently. And these are the few people who work with me so closely and having a lot of fun doing the work. Thank you.